Okay. Hello everyone, and happy to invite the many speakers to our sections, and happy that any participant to join this section today. And we are today the workshop we have nine speakers, and we separate two parts. The first part will be held by Gerald, and the second part will hold by myself. And in today's section, we will explore some genetic studies and some deep insights to both the marine or some other species. And all of these sections will focus on some application of the genomics, understand the evolutionary patterns and species history or genetic diversity. I hope that our speakers can share their some the research points in their work. I hope that everyone can get some good ideas and get some thoughts from our section today. So the first section will hold by Gerald, and there is five speakers. And yeah, Gerald, can you hear me? Yeah,
Yes, I do. Yeah. Okay. Thank
yeah.
you.
I think you can start. Yeah.
Okay. So I think
Okay.
Self will make a very good introduction of this session. I just make a little bit supplement to which I think this region of Earth is really exceptionally rich in biodiversity, featuring some of the most unique regions and environments, and also have a highest number of threatened species and impact by natural habitats and also exploitation of resources. Such that it's very important to understand the biodiversity in this region of Earth. So as Self mentioned, we have nine speakers. So with the further ado, we will introduce our first speaker. Professor Chihiro Kuraku, coming from the National Institute of Genetics in Japan, is going to talk about shark and ray genome sequencing on vertebrate karyotic organization. So without further ado, please welcome Professor Kuraku.
Hello. Some of you may regard sharks and rays just as a part of fish, but they belong to this distinct evolutionary lineage. Initially, we were driven by questions in evolutionary biology pursuing the vertebrate ancestor. But our motivation has been diversified. It now covers more topics. And to promote molecular studies, we run the consortium Squaremix under Earth Biogenome Project. And we aim at setting up genomic resources for addressing various biological questions. Here is a listing of the parties contributing to us, like aquariums and researchers. And here I send my best gratitude to them. Our activity is not limited to producing and analyzing sequences, and includes establishing analytical tools and materials, like culture cells and the protocol for preparing them. We also released a streamlined protocol, Squant Genome, to measure genome size without live cells. Our unique activity has been primarily enabled by these researchers inside my lab with the complementary skills. I welcome contacts from overseas researchers seeking opportunities to work with us. Now I get down to briefly introducing our recent achievements. Our survey of the members of the photoreceptor family opsins detected fewer gene repertoires in shark genomes, suggesting decreased reliance of sharks on vision and light. And we focused on this persistent member, rhodopsin, and reviewed a novel route for spectral tuning of the rhodopsin as a sign of adaptation to deep sea life. We also analyzed why sharks, unlike Eutherian momus, can sometimes switch between oviparity and viviparity, and reached one explanation that sharks have retained egg yolk proteins, vitellogenins, in their genomes, which is different from our genome. Here is a bridge to the main theme of the talk, karyotype evolution. Our analysis on opsins and heterogeneity in mammals revealed frequent gene losses like I showed in previous slides. And our genome-wide examination of gene loss highlighted an intergenomic heterogeneity of genomic sequences and other aspects. I relate this hypothesis with the karyotype evolution for the rest of my talk. The diversity of karyotypes revealed by cytogenetics is now accessible through DNA sequences. Here, the number of red lines show the haploid number of chromosomes, and the sum length of lines, genome size. With more species, we see taxon by taxon patterns. Sharks and rays show more lines than the others and gradual slope of line length. That's a typical trend of sharks and rays. Another trend is visualized here. Compared to the zebrafish medaka comparison, the shark-ray pair with the larger evolutionary distance shows more intensified signals of a chromosomal homology, like chromosome 1 to chromosome 1 and chromosome 10 to chromosome 10. The karyotype is really conserved even beyond large evolutionary distance. Here is a comparison between two batoid species with a significant genome size difference. The location of one-to-one orthologs shows more relaxed genomic landscape for the species with the larger genome, suggesting the elongation of intergenic regions. Here is a different view, zooming into several genes. Between the species, we see large intron size change, maintaining the relative location of exons. And genome-wide comparison shows quite variable patterns within fishes, as well as high resemblance between sharks and human with similar genomic organization. In summary, as far as we analyzed, large genomes of sharks and rays have longer intergenic and intronic regions, and this genome expansion were not achieved by whole genome duplication. Now, I talk about intragenomic variation. Sharks and rays have chromosomes of variable length within the genome. Along with chromosome length, GC content, gene density, synonymous substitution rate, and then tandem repeat density decreases, while gene length or intron length, as well as interspersed repeat density increase. A similar trend was reported for birds and reptiles previously. We tentatively grouped zebra shark chromosomes like this, but I especially have some opinions about the use of the word microchromosomes. Because there's no uniform definition in the first place. It would be safe to use the word microchromosome only within a karyotype in comparison with other with much longer chromosomes. Some studies present a conserved synteny in small chromosomes as evidence of their common origin at the common ancestor. But chromosomes intermittently undergo fissions and fusions, and probably there was a time with the microchromosomes after massive fissions and a time later or before without microchromosomes after massive fusions. These processes will be reversible in a bistable manner, and there will be no single answer. We found remarkable tendency, which roughly recapitulates what was previously shown for microchromosomes of birds and reptiles. Small chromosomes resemble the ends of larger chromosomes, and in every chromosome, the ends of certain lengths have such different characters from the rest. Inherently, short chromosomes are more widely occupied by its ends, and consequently, short chromosomes as a whole look more GC rich and exhibit other characteristics I mentioned. Our study also reviewed a shark 6 chromosome. We detected the chromosome 41 as a genomic portion with the doubled sequence depth for females, for zebra shark and whale shark. Genes on zebra shark X chromosome showed orthology to human and chicken genes on their autosomes, not sex chromosomes. I do not show here, but we could not find any commonalities with sex chromosomes of Therias species either. This suggests an independent origin of sex chromosomes between zebra shark and other species, other lineages. Notably, other studies provide more insights recently. The Nichols group in Belgium analyzed the genome size variation of sharks and found no trend in relation to karyotype evolution. Lohau's group in China showed an antiquity of shark X chromosome and also discovered Y chromosome for the first time. On the other hand, a collaborative study involving myself showed a distinct origin of a chimera X chromosome. So the chimera X chromosome may not be, homologous to Erasmo Blanc X chromosome. Here is a schematic showing homomorphy to heteromorphy spectrum of the sex chromosome pair evolution. As the Y chromosome found in the white-spotted bamboo shark from the Lohau's group is much smaller than its X chromosome and contain very few genes, I would place shark somewhere around here. Okay, I really thank you for your attention, and I would like to end my talk with the summary and these items that may assist your research.
Hi everyone, my name is Jessica Pearce. I'm a PhD student at the University of Western Australia and the UWA Oceans Institute. Today I'll be talking you through my research, reconstructing the tiger shark ancient demographic history through sequencing the first ever genome for this species and its family. But first, why do we care about sharks? Well, I don't know how many of you would remember this video that went viral earlier this year of a tiger shark chasing a baby turtle. Now, this demonstrates one of the key vital roles that sharks play in maintaining healthy ecosystem function through predation and top-down control. They're also a major source of income for fishing and tourism economies, and they're culturally significant here in Australia to many local Indigenous groups. But despite all of this, they're one of the most threatened taxa groups in the marine environment. From a genomics perspective, sharks are unique in their evolution. They represent an ancient lineage and have largely remained unchanged for millions of years, surviving multiple global mass extinctions. Given we're now facing another global mass extinction, information on past events and how species have responded could provide crucial insight for conservation. However, this class is severely underrepresented in genomics, with less than 1% having full genomes available. Now, tiger sharks in particular are an important species worldwide. They're distinctive due to their characteristic tiger-like stripes on their back and are distributed circumglobally worldwide in the tropics. They're also a keystone species for important ecosystems, such as the Shark Bay UNESCO World Heritage Area in Western Australia that was shown in that video. Worldwide, they've been targeted commercially and recreationally, but they're actually classified as near threatened by the IUCN due to a 30% global decline. And despite this, there is still much basic information that remains unknown about their populations, like their pupping and mating grounds. Tiger sharks are also unique as they represent the last of their kind, with all other genus and family members extinct. But despite previous genetic studies, which have shown two distinct populations, the Atlantic and the Indo-Pacific, to my knowledge, there has been no full genome sequenced for this species. And key characteristics, including genome size, chromosome number, and if this species displays heteromorphic sex chromosomes, were all unknown. Now, I don't have a lot of time to get into the methods here, but this is a brief overview of our process for generating a reference genome. This was all done as part of the Ocean Genomes Initiative following the VGP and EBP standards. To start, I collected a snap frozen blood sample from a male tiger shark and extracted high molecular weight DNA. From this sample, I've used PacBio long-read hi-fi sequencing, along with Illumina short-read sequencing and Dovetail OmniSeq Hi-C approach. I ran this combined data set through our in-house genome assembly pipeline with manual curation of the genome to assemble chromosomes and ensure there was no obvious misassemblies. This process then produced a final phased haplotype genome assembly with which I conducted the following analyses. To start, here's a quick overview of our tiger shark reference genome assembly with all quality metrics meeting the VGP standards. This assembly produced 42 chromosomes for haplotype 1, including an X chromosome. However, we've not yet been able to assemble the Y chromosome. To begin investigating the demographic history of tiger sharks, I looked at the heterozygosity and runs of homozygosity under the principle that longer runs indicate recent inbreeding, while shorter runs indicate historical inbreeding due to recombination. To put these results in context, I've included some of the same values for the great hammerhead, which is a critically endangered species with known levels of high inbreeding, and the short-fin mako, which has an ancient bottleneck but has since recovered. Now, as you can see, the tiger shark falls in between these two species for most of these values. This suggests that there has been moderate levels of historical inbreeding, as there's a reasonably high percentage of shorter runs, but there has also been some recent inbreeding due to the moderate proportion of longer runs as well. To investigate this further, I then reconstructed the ancient effective population size through a pairwise sequentially Markovian coalescent model. I ran this model with both haplotypes of this assembly, each with 100 bootstrap replicates, just to assess the difference. But as you can see, all of these have produced roughly the same results. Initially, this shows a high effective population size, or NE, at 150,000 individuals roughly 3 million years ago. However, this then declines slowly until 500,000 years ago, which then it appears to stabilize at roughly 20,000 individuals until a severe bottleneck occurs, dropping this to just 1,000 individuals from 2,000 to 7,000 years ago. Now, this shows that tiger sharks likely evolved during the mid-Miocene climate optimum, when the climate was even warmer than today's. It's interesting to note, however, that the first fossil evidence of modern tiger sharks dates back to 13 million years ago, so it's likely that we're missing a proportion of the early picture here. The slow decline seen corresponds to the beginning of the global cooling period and the start of the ice ages. As a tropical species, it's logical that the tiger shark would have undergone an adjustment period to a new global climate where many other species died out. Tiger sharks, however, did seem to stabilize through the rest of the ice ages that followed until the last one ended 10,000 years ago. What's most interesting here is that at this point, we would have expected to see population expansions with the beginning of the mid-Holocene warm period, But instead, what we actually see is this severe bottleneck occurring. Now, a previous study using a different analysis detected a bottleneck of Indo-Pacific tiger sharks around the same time, 2,000 to 3,000 years ago. Similar trajectories have also been found for white, short-fin mako, and scalloped hammerhead sharks with bottlenecks around the same time. But if the climate wasn't what caused these bottlenecks, then what other factors could have been in play? This also coincided with population declines in prey species, including sea turtles, seals, and whales, and the emergence of diseases following the last ice age has been proposed as a reason for these declines. But given sharks display heightened immune systems, this likely would have impacted the prey species, causing a food shortage rather than impacting the sharks themselves. It's also possible that global warming had other consequences and a flow-on effect with changing ocean conditions. The influx of fresh water following the melting of the ice likely caused changes in ocean currents and ocean chemistry that would have occurred and potentially impacting the species. With these results and the limitation of the PSMC model, it's hard to state exactly what caused this bottleneck. However, this evidence puts into question the implications for modern-day populations of tiger sharks. A shark bottleneck usually suggests reduced genetic diversity and capacity for future adaptation. The inbreeding resulting in the shorter runs of homozygosity that was detected likely occurred around 2,000 years ago, which matches our bottleneck timeframe. This level of historical inbreeding was also seen in the short-fin mako with a similar proportion and a steep bottleneck at the same time. But while that species appears to have recovered genetically, the tiger shark also displays moderate levels of longer runs of homozygosity, suggesting that there has been some recent inbreeding occurring in the past 200 years. But presently, there are no estimates for effective population size of the current populations, so it's unconfirmed if tiger sharks have actually experienced another bottleneck following this. I'm also in the process of a population-wide whole genome resequencing project to expand on these findings, which I hope to gain more conclusive answers to these questions, including a contemporary effective population size. Finally, I would just like to acknowledge the Minderoo Foundation for funding my research and the Oceanomics team for their help, along with my supervisors and my field team. Feel free to email me if you have any questions at the email you see there, and thank you for listening.
Okay, thank you. Thank you, Shuraku, and also Jessica Pierce from University of Western Australia to give talks. And Jessica just introduced the bottleneck in the tiger shark. So now we move to another group of species, which we're going to move to bird. So Katrina Sturt from School of Biological Sciences from University of Auckland is going to give the talk on the threatened bird species. So without further ado, Katrina, please.
Hi, everyone. My name's Kat. I'm a researcher currently working at Macquarie University, started a position here not too long ago, but the work I'll be presenting is from my postdoc over in Aotearoa, New Zealand, which is dealing with the role that structural variants versus single nucleotide polymorphisms can play in conservation genomics. So all of the work that I do essentially revolves around understanding adaptation and understanding it through the lens of genetics. We use genetics in all sorts of different ways in conservation and evolutionary biology. And what mostly underlies a lot of the work and analysis that we do is quantifying genetic diversity and the genetic variants that exist within a species. So for instance, we can have two populations and we can quantify the amount of genetic diversity that is within one population versus another. And from that, better understand whether one population is struggling and needs some kind of genetic rescue, or whether there's been a bottleneck or something else going on like that. Alternatively, we can have particular focal variants that maybe are associated with deleterious or even beneficial phenotypes that we want to track through populations. Or we can look at patterns between genetic diversity or specific variants and really key, for instance, environmental variables to understand how they may co-vary across a landscape. But all of these processes fundamentally rely on our ability to understand how the genome of one individual of a species is different from the individual of another individual in that same species. The way that we currently do this for a vast majority of the studies out there is through single nucleotide polymorphisms, or SNPs. These are single base pair changes that are different between individuals of the species. If we think about potential ways in which the genome of two individuals can be different within a species, along a continuum of sizes, SNPs exist at the very smallest end of that continuous spectrum. They are the smallest possible way that the genome of two individuals can be different. On the other hand, at the other end of the spectrum, we've got the very large changes. We've got the chromosomal or even whole genome duplication events. But for a majority of species, though not all, if we have an event in the genome that is that large, often it'll cause a difference between individuals that will mean that they're no longer compatible and no longer considered the same species. However, within this spectrum, there is a lot of different things, which largely fall underneath the umbrella term structural variants. What structural variants are is there are sections in the genome that are traditionally considered to be longer than 50 base pairs in length and can be up to a length of millions of base pairs. And they're chunks of the genome that have been deleted, moved, duplicated between individuals. So structural variants are largely considered to be very high impact variants simply because they affect more of the genome. And there's a big question whether structural variants reflect the same patterns as SNPs and vice versa. And for both biological and technical reasons, the answer to this is no. Structural variants often occur within regions of the genome that have really high recombination. And also, the existence of structural variants means that often our SNP calling can be a little bit stuffed up. And so we can't necessarily infer the identity of a structural variant based on the surrounding SNPs, which is a bit of a pain because in order to identify structural variants, what we often need is really high density whole genome resequencing, which is a very expensive way of going around to sequence individuals in your population in comparison to, for instance, low coverage whole genome resequencing or even reduced representation, which is what we often do to get SNPs. So we would like to ask the question is, if they technically don't resemble each other and we can't identify one from the other, does it really necessarily matter? Wouldn't we broadly see the same patterns and species across these two different markers? And that's exactly what we aim to test in the hihi. So the hihi, or the New Zealand stitched bird, is a species of bird that was once existing over most of the North Island of New Zealand, and now is basically only existing on a remnant population in Te Hatarua Toi. But from this location, they've been translocated throughout the country. And essentially, we're trying to reintroduce them to a number of different locations. Because of the interest in this species, a lot of work and a lot of man hours and a lot of money has been thrown at them. And so we have some really amazing resources. This includes a bunch of whole genome resequencing. In this case, for this project, we had 30 individuals with 20 times whole genome coverage, but we're really hoping to expand that in the future, and we're really starting to do that already. But in addition to this, we have some really cool biological data. We've got pedigree data for basically all of the individuals that exist. We know which individual has given birth to, or rather laid the eggs and raised up some individuals that have maybe themselves successfully raised the next generation. And so from that, what we can do is calculate individual fitness information, which allows us to then tie a really important phenotype, that of lifetime fitness, how many successful offspring has an individual produced, to the underlying genetic patterns. In this case, the patterns in the SNPs and the patterns in the structural variants. The questions that we were particularly keen on asking is around patterns of diversity and patterns of load. So the first question that we had was, do SNPs and structural variants occur in the same frequency in populations? And so we profiled the occurrence of the homozygous reference, the heterozygous, and the homozygous alternate across the population. And we found that there were differences in the overall frequencies between these two specific types of genetic markers, even though it's the same individuals. We found that SNPs largely occurred, were a little bit more frequent and occurred in the heterozygous state more often in comparison to SNPs. If we then visualized the same pattern, but instead we were looking at within individuals, where exactly the variants that they had within their genome, where exactly these variants occurred with respect to coding regions of the genome, so the protein coding genes. We found that, again, there were slight differences between SNPs and structural variants. In this case, we found that there was a lower likelihood of a structural variant occurring overlapping an intronic region, and a higher likelihood that it was going to be overlapping a gene region. And this reflects two key aspects of structural variants. Firstly, that they're larger, they're more likely to hit an important component of the genome, therefore more likely maybe to have a functional impact, a functional impact on the phenotype of the organism. And also, from this, we can maybe think that they're less likely to be neutral. So it appears that they maybe do occur in slightly different regions of the genome, possibly just due to their increased size, increasing the likelihood that they hit the coding region. And so what we did from this point is quantify something called mutational load. So load is the accumulation of deleterious mutations, and it's something that we're very interested in small populations like that of the hihi. So for this, what we did is we focused on the variants that occurred within these protein coding regions or directly adjacent to them. And we quantified the proportions of load within individuals. So in this particular study, we looked at load through two different lenses. First one is masked load, where the putatively deleterious allele occurs in the heterozygous state. The idea is that because it's being masked by the dominant and maybe not deleterious marker, the effect is not necessarily pronounced in the individual and thus is called masked load. On the other hand, if the putatively deleterious allele is occurring in the homozygous state, so the homozygous alternate state, what we have is realized load. And we contrasted the two different load calculations for SNPs and structural variants against one another and found that they were largely correlated across individuals. If you had high SNP load, you were more likely to have a high structural variant load. They weren't perfectly correlated. R-squared values weren't one, so they didn't 100% predict one another. And from this, we see important biological differences. When we get those structural variant and SNP loads, both the realized and the masked, and we correlate that with lifetime fitness that we obtain from the really impressive pedigree data that we have for the hihi, we found that overall, largely the same patterns were found across SNPs and structural variants. For realized load, the more realized load you had, the reduced lifetime fitness. And then the opposite then for masked load kind of being a little bit of a proxy for heterozygosity in functional regions of the genome. We saw that the greater the masked load was correlated then with an increase in lifetime fitness. And we see the same patterns across SNPs and structural variants, but importantly, you'll notice that the bottom two plots, structural variants, the slope of that regression line is a little bit stronger. And then what we did is, after analyzing these as individual models, we combined them all together to see if any particular aspect of load captured a unique component of genetic diversity. And what we found is that even though we have those same patterns in the singular models, together, what we see is that the only aspect of load that sort of captures a unique aspect and remained significant in a joint model was that of masked structural variant load. So that's not to say that the other components of load were not important. They were. It's just that it's what we're seeing here is that structural variant masked load contains a key aspect of genetic variation that was not captured in the underlying or the sort of basic SNP load calculations. And so what this tells us is that if we're particularly interested in conserving species and understanding exactly what types of genetic diversity drives important phenotypic trends like lifetime fitness, if we study only patterns that we find in SNPs, what we're going to be doing is missing really key and important aspects of genetic variation within the genome. And so we find then in the hihi, the key differences and the slight differences between the structural variant and the SNP patterns might be really key to allowing the species to be better conserved through the very careful management across its different populations. And so this study joins one of many that's showing that structural variants are capturing really important aspects of genetic diversity that were being used and applied in all sorts of conservation evolutionary genomic settings. And thus, hopefully, will continue to be explored in lots of different systems and applied in all sorts of different ways with conservation focuses and also to understand better how evolution can happen because of these large variants, in addition to the traditionally studied SNPs. I'd like to thank you all for listening, and also thank my boss at University of Auckland who worked with me on this project, Anna Sancho, and welcome any questions you may have.
Okay, thank you, Katrina, for the talk. It's very interesting about the whole genome resequencing and comparison in between the SNPs and also the structural variation. I mean, so in birds. So again, we move from shark and ray to bird, and then to another group of interesting animals. And now we are going to see another talk going to be given by Ashwini Loganathan, coming from Monash University of Malaysia. So she's the speaker is going to talk about the situation in endangered hedgehog and also seahorses. So without further ado, I mean, we have now next speaker. Thank you.
Hello everyone, I'm happy to participate in the Biodiversity Genomics 2024. So I will start with a brief background on myself. So I'm Ashwini, I'm a research fellow at Monash University Malaysia Genomic Platform. So my research focus is generating high-quality reference genome assemblies using high-throughput sequencing technology. So my aim is to draw attention on the conservation of endangered animals in Malaysia through genomics. I'm also responsible for generating a generalized bioinformatic pipeline that can be used for genome analysis, such as mapping, assembly, variant calling, 16S rRNA, and shotgun metagenomics, among others. So these pipelines are used for both internal and external collaboration and services, as well as for students' project. So we are currently in the midst of setting up the Malaysian Biogenome project, which aims to generate high-quality reference genomes to capture eukaryotic genomic biodiversity in Malaysia. So this is to expand our collaboration with the Department of Wildlife and National Park, which is also known as Perhilitan. We also aim to become a part of the Earth Biogenome Project and the Vertebrate Genome Project that aims to sequence, catalog, and categorize the genomes of all of Earth's eukaryotic biodiversity. So these are some of the species that we are currently working on under the Malaysian Biogenome Project. So we have the seahorses, we have the hornbills, the Malayan tiger, we have the Malayan sun bear, the Malayan tapir, the samba deer, the gao, the dole, and sea cucumber. So currently all of the samples were provided to us by the Department of Wildlife and Nature Park. So today I would like to share one of the ongoing topic entitled Reference Genome for the Endangered Malaysian Hedgehog and Tree-Spotted Seahorse. So this project is a collaboration between Monash University Malaysia, Monash University Australia, and our NGO Save Our Seahorses. So Malaysia is an extremely biodiverse country and is fortunate to host 12 of the 46 species of seahorses found globally. So the high demand for dried seahorses for use in traditional Chinese medicines has resulted in overfishing and exploitation, which has subsequently led to an alarming decrease in seahorse population. We propose to use high-throughput sequencing technology to generate reference genome for the endangered hedgehog and also the tree-spotted seahorse. So I chose these two species because they are commonly found in the traditional medicinal shop in Malaysia. So on the left over here is the hedgehog seahorse, and on the right over here is the tree-spotted seahorse. The primary aim for this project is to generate two high-quality reference genomes for the Malaysian hedgehog and tree-spotted seahorse. And in the phase 2, we like to identify genetic variants that differ between these two species, which will be then used to develop a genome-wide assay for discriminating wild population of the species. Additionally, we aim to use species distribution model to highlight the effect of climate change on species distribution and identifying global biodiversity hotspots. So for today, I'll be just focusing on phase 1 of the project, which is to generate reference genome for the two species. So this is the geographical area of the study. So the samples are actually a fishery bycatch, which we obtained it from our local fishermen. So samples were collected from Endau Johor, which is the southern part of Malaysia, and also from Hutan Melintang, Perak. So once we receive the sample, we proceed with the high molecular weight DNA extraction. So since these are fishery bycatch, by the time we receive the samples, they were no longer fresh. So it took a while for us to actually get the high molecular weight DNA and then proceed with the library preparation using the SmartDel Express template prep kit. And then finally, we did our sequencing using the PacBio Sequel 2E. Once we have the raw data, we proceeded with the bioinformatic analysis. So basically what I've done so far is that I did the genome profiling, and then I proceeded with the de novo genome assembly. So I've used 4 different assembly tools. So Canoe and Highfism are most widely used, and I've also included WTPG2 and IPA from my literature search. And then I proceeded with the quality assessment of the genome assembly. So these are the raw read statistics. As you can see, for the hedgehog seahorse, the number of reads is around 618,000, and for the tree-spotted seahorse is around 281,000. For the read length, for the hedgehog, we have around 4 billion, and for the tree-spotted seahorse, we have around 3 billion read length. As for the BUSCO assessment, for the hedgehog seahorse, as you can see, Canoe and WTVG gave the highest score with 91.1% for the Canoe and 90.2% for WTVG. Similarly, for the tree-spotted seahorse, both Canoe and WTVG gave the highest score with 85.8% for Canoe and 89.4% for WTVG. As for the assembly statistic, we can see that for both Canoe and WTVG, we have the lower number of contig and larger N50 value. Right? So this is all that has been done so far. So next, I'll be doing the polishing. And also we have just implemented Galaxy on our HPC. So I will download the VGP pipeline by Ria et al. And run the assembly pipeline on Galaxy as well. So Hi-C library preparation is also currently ongoing. So for now, there are so many uncertainties because we haven't done much yet. So hopefully once we completed our Hi-C, we can get more input or details on the completeness and also the quality of the assembly. I think that's all from now. I'd love to hear your feedback, your comments, or any advices regarding this project. Thank you.
Okay, thank you, Ashwini, for sharing the progress on the situation on the hedgehog and seahorses genome assemblies. So after 4 wonderful speakers, I mean, talking about from shark ray and tiger shark, bird, hedgehog, seahorses, we're going to switch the gear a little bit. And now we're going to talk about the next speaker, the 5th speaker, is going to talk about genome assembly methods. Richard Evers, coming from the University of Western Australia, is going to share with us his research. So without further ado, Richard, it's all yours.
Hi, so I'd like to just thank the organisers for the opportunity to talk today. And today I'm just going to be talking about some of the work we've been doing using nanopore reads to try and improve some of our hi-fi assemblies. I'd like to just start by acknowledging the Whadjuk Noongar people as the traditional owners of the land on which I've done this work and recording this video. The Whadjuk Noongar remain the spiritual and cultural custodians of their land and continue to practice their value languages, beliefs and knowledge. I'd like to pay my respects to the traditional owners past, present and emerging of the lands on which I live and work in Western Australia. So just to give you a bit of background on what I'm trying to do here. So I'm part of the Ocean Genomes team. I'm not going to talk too much about ocean genomes as other members of the team are giving presentations in the conference. But suffice it to say that we're making a large-scale reference genome library of marine vertebrates. This is aligned with the Vertebrate Genomes Project and as Biogenome Project. And so the goal is to make phased chromosome-level assemblies using their standard protocols and standards with Hi-Fi and Hi-C data. What we're trying to do here is investigate if and how adding nanopore data can improve our assemblies. So previously tried some gap filling, which I'm not going to talk about with mixed success. And one of the issues there was potentially a downgrading quality. But we've also been struggling a bit sometimes with some of the samples in non-model organisms to get the really, really long reads needed for ultra-long sequencing. So what we're asking here is, can we just use the regular LSK nanopore data and feed that into the Hi-Fi as an assembly using the ultra-long parameter. And at the same time, trying out just the new telomere mode to see if that improves our telomere recovery. And the general approach is assemble the data with Hi-Fi and Hi-C, then assemble the same data, adding 20 kb plus nanopore reads with the ULK setting in Hi-Fiasm, and then again with the telomere mode. So just one thing I want to point out is we have filtered all the reads down to a minimum of 20 kb, but they're not true ultra long, but they are longer than most of the Hi-Fi reads would be. What we're using for this sample, this study is 20 bony fish from the Ocean Genomes project. These all have Hi-Fi, Hi-C, and nanopore data with a range of depths and a range of sort of length profiles and things. Not going to say any more about that now, but happy to take questions. In terms of what we're actually looking at, so essentially the way that the study is working is all the assemblies with the addition of the nanopore read are being compared to the same data assembled without the nanopore data, looking at absolute and percentage improvements or indeed decreases in a variety of statistics. And because the phasing isn't always consistent, we're not comparing HAP1 with HAP1. We take the best haplotype for each metric for each assembly and compare those, and then take the worst ones and compare those. I'm not going to go into all the details in the interest of time. Have about nearly 50 metrics in the general categories one would expect, looking at assembly quality, so contiguity, completeness, some quality metrics, a little look at repeats, and then telomere recovery. And then done statistics on a core set of 20 of these to reduce the multiple testing. So the main take-home message, as one might expect, but it was a pleasant surprise given that we're only giving reads of 20 kb plus, is that when we add the nanopore reads using the ULK setting, we get a significant improvement for 8 out of our 20 core metrics. And basically, these are all the metrics that are connected to improved contiguity of the genomes. So on the right here, we have a heat map of the 22 different fish, best haplotype and the worst haplotype. Red is good, that's an improvement in statistic, and blue is bad. So don't need to worry about the details. If you see a lot of red, then adding the nanopore data has made things better. A lot of blue, it's made it worse. And the main thing is we've got a big reduction in the total number of contigs, a fairly big increase then in the contig N50, and that translates into the proportion of the genome on sort of chromosome scale contigs. And some improvement, although it is variable, was seen for every fish in our sample. The completeness of the assemblies isn't altered very much. Fusco and Mercury, both with an odd exception here, this outlier, which I can take questions on, essentially don't change very much, but that's not too surprising because our basic assemblies were extremely high quality and high completeness. There is some evidence based on read depth profiles that adding in the nanopore data has removed some of the lower quality sort of false duplicates and things that may be hanging around. And so there is a general reduction in the proportion of the assembly which has low coverage when we map the reads back on. So that's another sign that maybe it is slightly improving the quality as well as the contiguity. As an example of just seeing how it's handling the sort of long repeats, which one might want that long reads to do, we looked at the recovery of full-length ribosomal DNA units repeats, which are hard sometimes to assemble. There's a bit of a variation across the samples, but overall there was a reduction in the number of unplaced contigs that are just purely ribosomal DNA, and a slight increase in the number of full-length ribosomal DNA units which get assembled into chromosome-level contigs. Not going to talk too much about the telomere mode, but suffice it to say that switching on telomere mode did indeed increase the number of telomeres, and that's true across the board for all our samples. And this resulted in a slight increase as well in the number of telomere to telomere gapless contigs. We're obviously interested in trying to then work out, well, if this is making an improvement, why, what's important. So in order to do this, we had a strategy where we took the contigs. So this is a synteny plot of the HiFi contigs. In the middle, we have the HiFi plus nanopore. Basically, what we did here was reference scaffold the HiFi off the nanopore addition assemblies to try and see how these things were going to go together. So this is using a program called ragtag. Then mapped the gap. So if we put two contigs together, there's a gap in the assembled contig. Mapped where that was onto the assembly that had the addition of the nanopore, so the more contiguous assembly, and then looked at what was going on there in terms of reads and mapping onto this contig from the HiFi plus HiC plus nanopore assembly. Hopefully that's clear. What we then did is took a set of control regions, essentially from the alternative haplotype in each case. Mapped on then the HiFi reads and the nanopore reads, and looked at a couple of things. So one was the depth profiles across these regions, and one is how many reads were actually spanning that entire region, plus a bit of flanking sequence. Again, don't have time to go into the details, but the take-home message is that there seem to be overall, so this line is if the gap regions and the controls are the same, and again, we've got our blue and red. For the HiFi data, there seemed to be a big reduction, so it's all blue, and there's a big loss in coverage for these regions, whereas for the nanopore data, it was very similar. So there seems to be something going on here where the reason the nanopore data is improving it is because it's hitting parts of the genome that didn't have good coverage in HiFi reads. When we looked at the length, there wasn't any obvious pattern where more longer nanopore data was actually helping more. It just seemed to be about the sort of the amount of data we were adding in. So there's some questions then of why this is. So what are the next steps? So none of these were taken up to the actual full chromosome level. So the next steps is to actually then compare what's going on with some of our genomes which have been curated up to chromosome level and see whether the improvement follows through, and try to identify those regions a bit better, and then work out what's actually driving this. Is it just adding more sequence? So would increasing the HiFi depth have the same effect? Is it that there's a different way the nanopore data is incorporated with this mode, which means you don't need to add quite as much to get the same improvement? Or is there actually some difference in bias in the technologies, which means that there are regions of the genome dropping out of the HiFi more than we would expect? Then we'd like to go to more complex organisms, like sharks that have bigger, nastier genomes than the fish. So with that, I'd just like to acknowledge the Minderoo Foundation for the funding, and particularly the chairs, Andrew and Nicola Forrest, and then give a shout out and thanks to all the people at the Minderoo Oceanomics Centre at UWA, where I work, and in particular, the Ocean Genomes team, some of whom are speaking in other talks, all of which contributed to this work. And I'll be happy to take questions in the question session later, or feel free, please, to email me. Thank you.
Okay, thank you, Gerald, for the hosting the first part of these sections, and the next part of section will still focus on the evolutionary genomics and ecological dynamic across different species and ecosystems. And right now, in the second part, there is 4 speakers there, and we'll focus on how genetic data will be used to understand the ecological interactions and evolutionary process. So we're going to invite the first speaker, Dr. Emmer, from the Ocean Omics Center, University of West Australia, and will start his work and discussion on the high quality genomes in Australian Legionnaires species and highlight some these parts of the genetic resources for sustainable the beneficiary management. Also, okay. So happy to Dr. Emma.
Okay, hi everyone. My name is Emma de Jong, and today I'm going to talk about some of the work that we're doing to produce high quality genomes for some Australian Legionnaires species. I would like to begin though by acknowledging that the work that we do takes place on the land of the Whadjuk Noongar people, and to pay my respects to the Whadjuk Noongar people in recognition of their ongoing connections to land and sea country. So first, I just want to introduce the Ocean Genomes team and describe the work that we're doing more broadly. So we are situated in Perth on the west coast of Australia, and our aim is to accelerate and scale the production of reference genome resources for marine vertebrates with a focus on quality, scalability, and accessibility. And one of the reasons that we're doing this is to help facilitate environmental DNA monitoring applications. So we work alongside another team focused on using environmental DNA from water samples to monitor ocean biodiversity. But to do that effectively, we need a comprehensive library of reference sequences. And in addition, we recognise that these genomes will be foundational resources to inform biology and conservation management as well. So one of the first things that we've done as a team is to first conduct a review to assess how many marine vertebrate species currently have a reference genome available on NCBI. And this figure is showing data that we pulled together from last year. And we found that over 96% of taxa are lacking representation. You can see that this does vary across the broader taxonomic groups, where the least representation is on the right here for ray fin fish and for cartilaginous fish. So we're working to try and fill some of this data gap with a focus on the Indo-Pacific region. And so in this talk, I'm going to highlight one family in particular that we have quite a few representatives for. So that's the Legionnaires family, also known as snappers. So these fish are found globally in tropical and subtropical marine waters. There's an estimate of 113 species worldwide, more than half of which can be found in Australian waters, yet only 2 species currently have a chromosome level reference genome assembly on NCBI. And these fish are an important group because they're highly valued commercial and recreational fisheries species. They're also great candidates for aquaculture. Some of these species are already being farmed in Asia, and this is likely to continue with increasing global demand for sustainably farmed seafood. So with this project, we're aiming to address the data gap by producing high quality reference genomes for 20 different Australian snapper species, and then using these reference genomes to comprehensively map the immune gene repertoire. So this is something I'm really interested in, and this information is really important for understanding disease vulnerability and the impact of environmental stress, both of which come into play into aquaculture systems. And so how are we doing this? So we're producing our reference genomes with a combination of Hi-Fi and Hi-C data for each individual. And we're following this workflow here, which has been developed by the Vertebrate Genome Project. And so without going into too much detail, essentially we're just trying to meet all of the metrics for high quality reference genomes set by the VGP and the Earth Genome Project as well. So to date, I've generated the reference genomes for 14 Legionnaires species plus 1 close relative. So it's important to have this one in the analysis so that we can infer evolutionary relationships properly and provide context for gene evolution. We also have several other species in progress, and we should have the genomes for these completed in the next few months. And so just to describe briefly what our reference genomes for the Legionnaires species are looking like. So the median genome size is just over 1,000 megabase pairs, and that's the box plot here summarizing that data. And they typically have 24 chromosome pairs for 2n of 48. And in terms of the data coverage that we have, so we have a median of 53x for Hi-Fi and 62x for Hi-C. So we have quite good coverage for our genomes. And fortunately for us, most of these fish assemble really nicely. So I'll just show you some statistics now about our assemblies. So in terms of contiguity, we're able to achieve quite high scaffold N50 values. So the target here that we're trying to achieve is 20 megabase pairs, and all of ours are sitting above 40. They also have relatively few gaps. So that's this plot on the right here. Our target here is for less than 200 gaps per gigabase pair, and most of ours are achieving that. So these red dashed lines represent our target values to be aligned with the VGP and EBP metrics. In terms of BUSCO completeness, here we're aiming for at least 95%, and you can see that we're achieving 99% or greater. So this indicates that our reference genomes contain almost all of the roughly 3,500 highly conserved genes that are present in all ray-fin fish. And then finally, looking at correctness, without going into too much detail, we can assess the assembly against the raw read set and then evaluate the error rate in the assembly. And so here we're trying to reach a target minimum of 50 as our QV score, which is equivalent to 1 error in every 100,000 base calls. And you can see that we're achieving above that as well. So next, I just wanted to present an overview of the methods that we're using to characterize the biology of these genomes. I want to emphasize here that I'm relatively new to this kind of analysis, so any feedback and suggestions are more than welcome. So following on from manual genome curation, I've run repeat modeler to characterize the repeat content. I'm not showing any of that data today, but I just thought I would mention it and also say that we've left our genomes unmasked for the annotation step. And so regarding the annotation, here I've chosen to use Gemoma for a homology-based annotation, for which I have leveraged the reference annotations from 3 model species, so the zebrafish, the clownfish and the fugu or the pufferfish, plus 1 close relative for which a reference annotation was available. The output from Gemoma was then fed into AutoFinder to identify gene families and author groups. And from here, I'm aiming to characterize the biology of these gene families and also assess the gene family evolution between species as well. And so here is some preliminary data coming out of AutoFinder. So this plot shows the number of groups, number of author groups, sorry, on the y-axis and the number of species that have that author group as part of their genome along the x-axis. And the takeaway here is that most author groups identified are shared across the Lugenidae species as well as our outgroup species, while there are some that are unique to each species as well. And another way we can assess the output is by looking at the species tree based on this data. So this is a preliminary tree that has been inferred from all of the author groups. And this species at the top is our outgroup. So it's nice to see that one sitting there. And another thing to note is that all of the Lucanus species are grouping together as well, which gives me some confidence in the analysis. And then finally, the next step is to look at which author groups are expanding or contracting across the tree. So for that, I've used CAFE5, which takes in the tree from AutoFinder and account matrix of all of the species and all the author groups contained in their genome. And so here at each node of the tree, I've put in the numbers for how many gene families or author groups are significantly expanding in red or contracting in blue. So there is a lot that we can look at here. I'm quite interested in what's happening at this sort of part of the tree here. So this is the branch point that contains all of the Lucanus species separately to the others. So that's something I want to look into a little bit more. Yeah, just to finish up, just to briefly talk about the next step. So like I said, this is all preliminary at the moment. We're still producing some of the reference genomes that we want to include. So in terms of next steps, I do want to go back and refine the genome annotation workflow to possibly include some RNA-seq data. So we have sampling and data production for that is ongoing. And then secondly, I also want to establish some workflows to characterize the gene families or author groups and their biology at a more systematic level, especially since I am most interested in the immune gene repertoire. So suggestions on ways to do that are also more than welcome. Okay, and I just want to end by acknowledging the whole team and all of our partners. Yeah, this program is a really huge effort contributed to by a lot of different people. So yeah, just wanted to acknowledge everyone here and thank you for listening.
Okay, thank you, Amos. I think that the research is can provide a valuable genetics resources to enhance maybe some knowledge of the gene family and benefits of the broad conservation efforts. Yeah, so we're gonna invite the next speakers, Dr. Alex from Charles Darwin University, and he will discuss the spatial population dynamic of decline in some mammals in the wild-prone landscapes. And he will also highlight some how the genetics can aid the conservation strategy in Northern Australia. So welcome, Dr. Alex.
Hello, I'm Alex Carey. I'm a PhD student at Charles Darwin University in Darwin, and I'm going to talk about my research into using genetic data to understand species responses to fire. So I'd first like to acknowledge the Larrakia people and the Tiwi people and pay my respects to their elders past and present. So in recent decades, populations of native small mammals have been disappearing across mainland Northern Australia, and they've become restricted to coastal areas and islands. So the Tiwi Islands here are important refuges, but in recent years on the Tiwi's, Melville Island, the bigger island here, these declines have been documented as well. So what's driving these declines? The primary driver is predation by feral cats, but this is made worse by large and intense fires and feral herbivores opening up the landscape and increasing the predation efficiency of cats. And so to manage this, fires are lit in the early dry season to reduce the size and intensity and leave some refuge areas for small mammals. And whilst this has been done, we haven't yet seen conservation outcomes for small mammals, and this is potentially because we don't understand how species respond to fire events and persist in fire-prone environments. So on their own, we have a decent understanding of landscape dynamics, of fire regimes, and habitat succession, as well as population dynamics, so how species disperse and their life histories. But we don't understand quite well enough is how populations are able to persist in fire-prone and disturbance-prone landscapes. And genomics can provide unique insights into dispersal and population dynamics at both fine and broad scales. So the species I'm interested in are the northern brush-tailed possum, which is an arboreal species, relatively long-lived, low fecundity, low dispersal, and then comparing that to the northern brown bandicoot, which is a ground-dwelling marsupial, lives for just a few years and breeds faster than rabbits. So the two objectives I have is looking at the short-term responses, so using a fire experiment to understand how these different species respond to and recover from a fire event in areas of low and high fire frequency. And then in the longer term, using a broader landscape genetic study to look at what is driving what landscape variables and environmental variables are driving these genetic connectivity patterns. So for part one, the fire experiment, we have these two areas of low and high fire frequency. And then within each area, we have 8 plots, 3 of which were burnt and 3 were unburnt controls. And then we trapped before fire, after fire, and 1 year after fire. And we hypothesized that patterns of genetic diversity and relatedness will be different between these species and between these areas of fire frequency. So this first result is mean site relatedness. So you can see here for bandicoots, in each session, mean site relatedness was constant, so it remained the same in both the high and low fire frequency area. But for possums, we had an increase in the low fire frequency area, but constant in the high fire frequency area. And then in this analysis, we identified related pairs, so kinship pairs, and then measured the distance between them in each session. And so we found for bandicoots, the blue lines here, the distance between relatives increased in the post-fire session, but for possums, the red lines, they remained consistent in each session. But fire treatment itself was not included in this analysis. So some quick conclusions of this part. We found more pronounced effects in the low fire frequency area, differing responses by species reflecting their different recovery strategies. And then this nuanced ecological information is required for effective fire management for conservation. So then looking at the broader scale across these 2 islands, we collected all these genetic samples over the 5 years. And then just some quick context, Bathurst Island, the smaller island, threats are largely absent, so no cats or buffalo. Populations are stable and it receives higher rainfall, so higher productivity and habitat succession after fire. Whereas on Melville Island, they have cats in high density and buffaloes, and areas have been cleared for plantations. They have declining populations and lower rainfalls. So we have our genetic patterns across the islands, and we want to look at these different landscape layers and understand if they're driving these patterns. So we're looking at cat and buffalo, 3 different variables of fire reflecting frequency and intensity, and then vegetation type and rainfall, topographic features, distance to coast and water, and then our null and geographic distance layers. So we optimize each of these layers till they best represent genetic distance. We also combine the layers and optimize them again to see if they can better reflect genetic distance. And then we take each of these surfaces and model them and bootstrap them until we get a consistent outcome as the best description of genetic distance. So which of these landscape characteristics on their own or in concert are driving genetic structure patterns? So for bandicoots on Melville Island, the best selected model was topographic ruggedness. And interestingly, it's the low topographic ruggedness areas that's highly resistant to movement, whereas the high topographic ruggedness facilitates movement. This could be an indication of habitat complexity, as this is the most fine scale layer we have, and that could be a refuge from the threats. And then we compare that to Bathurst Island, the island without the threats. The best selected model is geographic distance. And so that lines up with what we found in our genetic structure patterns. Everyone's fairly similar there. Possums on Melville Island, again, it's topographic ruggedness, but this time in concert with fire frequency. And so interestingly, the fire frequency layer, it was the high fire frequency that was actually facilitating the most movement and connection. So perhaps the fire frequency is necessitating movement here. And then for possums on Bathurst Island, it's the high rainfall in the north here that was the most resistant to movement, but perhaps as a more sedentary species. The possums, this high rainfall, high productivity, more stable resources meant they didn't have to move quite as much. So some conclusions from this part. Genetic diversity and structure patterns are different between species and islands. The drivers of genetic patterns are associated with how landscape characteristics interact with species traits. And I think it's interesting that the threats, so the island that has feral cats selects the surface that's the most fine scale and complex, whereas the island without the threats is selecting either geographic distance for the more dispersive species or rainfall for the more sedentary species. And so this complexity of habitat may be important in landscapes where threats are present, and high fire frequency itself is not reducing gene flow. So looking at both of these studies together, found a dispersal response to fire events detected in bandicoots, but not in possums. And differing responses by species are reflecting the recovery strategies. And then in the longer term, fire frequency itself is not reducing population connectivity, and the drivers of population connectivity vary between species and islands depending on the species traits and the landscape context. So these genetic insights go beyond the scope of other methods and are crucial to understand and improve management. Tiwi mammals have pre-adapted traits to persist in frequently burnt landscapes. Fire management needs to be place and species specific. And lastly, cat management is crucial. Thank you, and thank you to all these people.
Oh, okay. So, thank you, Dr. Alex, to share the findings of the fire experience, and then Alex gave some genetic studies of the to reveal the population recovery process and its impact. So we got to invite the next speaker for the Nanya Technological University, Dr. Jiao, and he will discuss the evolutionary ecological of Southeast Asia, the rainforest flora, and focus on the genetic diversity and adaptation of tropical plant species. So welcome, Dr. Jiao.
Thanks for giving me the talk here at the Biodiversity Genomics conference. So I'm talking about the genomic perspectives on Southeast Asian rainforest flora. So I'm from Finland myself, and then in Finland we have about 1,300 species in the entire country. But in Southeast Asia, so there are over 50,000 species that have been recorded in that region. So there's a massive amount of biodiversity that's not just present in the boreal regions. So there are several different hypotheses why the biodiversity is so high in the tropics. So one of them is that there's this cradle of life hypothesis that all the biodiversity has been born in the tropics, in the aseasonal tropics, and then the species have occupied their niches there, and then eventually migrated out to the boreal and more seasonal environments. So most of the plant orders have never wandered out from the aseasonal tropics. There's also suggested that there is this positive feedback loop on the biodiversity. So if you already have a high biodiversity, then it creates more slots, more niches for new species to evolve. Also, it's linked to this co-evolution. So now that the species are there, then they are interacting, and then this co-evolution is also creating more niches for different species. So symbiosis, or then pathogen host interactions. In Singapore, if you look at biodiversity, there is very little of rainforest left. So in the heart of Singapore, there is about 2 square kilometers in Bukit Timah Nature Reserve, where there's about 2 square kilometers of primary and secondary rainforest that was conserved in 1850s. So about 1,200 angiosperm species have been identified there over time, but many of them are already extinct locally. So we started sampling the biodiversity here in Bukit Timah in 2018. And then we went forward and then sequenced all the angiosperm species that we encountered there. So 3 years in, we had collected about 547 plant species, and then we went on and sequenced those. And then out of those, we received about 499 good quality genomes. So we still continued collecting the samples. So we now know that there are altogether 882 species in the entire Bukit Timah. So now we can say that we sampled about 60% of the entire angiosperm biodiversity in the rainforest. And it represents about 80% of all the large trees, for example, at the site. Then we went on and analyzed the entire plant assembly altogether, and we didn't find, surprisingly, that many polyploid species in there. So polyploids are one of the suggested main mechanisms for sympatric speciation. So you would have expected that there are more polyploids in the tropics, but then that wasn't the case. We didn't find even old whole genome duplication events in here in Bukit Timah, so you couldn't explain the biodiversity with that either. And then we didn't find any hybridizations between species, so even the hybridization didn't explain the biodiversity. So possibly these deleterious effects of polyploidy have made it very difficult for the young polyploids to establish in this highly competitive environment. Then we looked at the phylogeny of Bukit Timah and then identified that most of the biodiversity already dates back to Cretaceous period. So the ecological niches apparently were occupied already during the Cretaceous seasonal rainforest environments. When we did protein clustering for all the predicted genes in the Bukit Timah plants, we found that this proteome space didn't saturate. So every species had their own set of genes that contributed to the plant assembly. So apparently this supports this niche theory for every plant. So every species would occupy their own specific niche. A principal component analysis grouped all the plant orders together, and then the main principal axes were associated with secondary metabolisms and defenses. So our hypothesis is that this secondary metabolism and symbiotic interactions, co-evolution between different species, then is a cohesive factor between the plant orders, and so it dates back to the Cretaceous period. But then they are highly diverse with respect to the secondary metabolism and the interactions. At more recent timescales, when we looked at differences between species, the species-specific gene families were enriched for resistance genes, so which provides support for this hypothesis that the pathogens are driving this speciation in the rainforests. We looked more deeply into R gene counts, and we found that it's correlated with the lifespan of the trees or of the species. So the longer you live, the more resistance genes you have there. So apparently it's how much you emphasize the defenses in your life strategy and lifestyle. We then went on and looked at community level effects also in the forest types. So the Bukit Timah is very well studied, and then we have GPS coordinates for all 10,000 big trees in the Bukit Timah. So now we can connect these coordinates with the actual genomes of the sequenced plants and then look at if there would be some stratification between the primary and secondary rainforests. What we found is that the resistance genes seem to be enriched in the primary rainforest regions. So the plant assembly there emphasizes this stress tolerance and pathogens defenses more than in the secondary rainforests, where the plant strategy is to flower as soon as possible and produce progeny. So the lifestyle affects the R gene counts in the genome. We did the same thing for cytochrome P450s to look at the diversity of secondary metabolism in the forests as well, and found that this diversity is surprisingly lower in the primary rainforests, and then in the secondary rainforest there is much more cytochrome P450 families than you would expect by chance. When we zoomed into the different cytochrome P450 families, we find that there are different cytochrome families enriched in different forest types. So which suggests that the two forest types are sharing having a different kind of chemistry. So in primary forest, the cytochromes that were enriched were mostly associated with plant defenses and secondary metabolism, whereas in the secondary forest there are also other strategies that are present. So also defenses, but then also strategies related to faster life cycle. Finally, then we looked at this demographic analysis by looking at the plant histories from all of these species that we sequenced, and found certain turning points in plant evolution. So for example, we found a peak in high growth rates during the Eocene, when the forests were highest about 30 million years ago. Then where we had found also a peak of decreases around late Miocene cooling period, and then more recent times we found mostly decreases in the forest sizes, so which tells us that the rainforests have not been expanding lately, and then apparently they have not been reacting to this interglacial fluctuations during the last 2.5 million years. So apparently the forests are not reacting fast enough to actually occupy the land that has been uncovered during the glacial maxima. So here we provide this first view on ecosystems. So we coined this ecosystems genomics. So, and we show that this sequencing of a rainforest is feasible and it can be done, and it provides new complementary view into ecology, and it provides a view into past, present, and future of tropical forests. Finally, thanks to everybody. So I was Jarkko, and then thanks for Yunjing and JJ Jiajun Yam, who were the first authors together with Matti Niissalo, and then also the collaborators. Thanks.
Okay, thank you, Dr. Jarkko. And happy that he can bring some valuable insights for this, maybe some unique ecosystems biodiversity. And right now we will welcome to the last speakers, which is from the Institute of Evolutionary Biology in Spain, the Dr. Gabriel, and he will present his research on the chromosome level reference genomes and some medically significant and Arabian homed wafers, which will maybe I think that you comes our understanding and evolutions and species conservation. So welcome, Dr. Gabriel.
So hello everybody, my name is Gabriel. I'm doing my PhD at the Institute of Evolutionary Biology, and today I will be talking about snakes and specifically about venomous snakes. So if we check a quick phylogeny of snakes, we can see that we have blind snakes and pythons as boas as basal groups, but within the Cynophidia group, here shown by the red dot, we have all the venomous snakes. We have some iconic groups as vipers or elapids, but we also have other maybe less well-known groups of venomous snakes as atractaspids or colubrids. And today we will be focusing on vipers. So there are more than 380 described species of vipers, and they are widely distributed in America, Africa, and Eurasia. There are two main subfamilies. Here in the phylogeny you can see them, Crotaline on the top and Viperine on the bottom. And the interesting feature of this group is that most of them are highly venomous to humans. And today we will be focusing on a small genus within the Viperine subfamily that is called the genus Therastes. The genus Therastes has three main species, Therastes therastes, Therastes viper, and Therastes gaspereti. The first two are present in North Africa, while the third one is endemic to the Arabian Peninsula. And today we're going to be focusing on this third species, Therastes gaspereti. So this species is endemic to the Arabian Peninsula and is adapted to arid and hyperarid environments. But why we're focusing on this species? So we are currently working at the genus level, and we are producing several genomic analyses and studies on this genus. In fact, we recently published a paper talking about the evolutionary history of this genus, but then we realized that we needed more genomic resources in order to keep exploring the evolutionary history of this genus. Moreover, as I told you, this species is also venomous, and we also wanted to study their venom from a genomic perspective. In fact, venom evolution has traditionally been studied mainly focusing on a proteomic or a transcriptomic approach, but when researchers have incorporated genomics into the equation of venom evolution, they have been able to address other questions as they have been able to identify the different toxin genes found in the genome, or they have been able to compare gene copy number variation of toxins among species. So for that, the main objective of our work is to produce a high-quality chromosome-level reference genome for Therastes gaspereti, and then use that reference genome in order to identify the different toxins at the genomic level following a multi-omic approach. So for doing that, what we did is to sequence long-read data, linkage data, and Illumina data in order to produce an assembly. We sequenced RNA-seq and iso-seq from different tissues of one individual in order to do the annotation, and then we extracted the venom in order to do the proteome. So we managed to assemble a high-quality chromosome-level reference genome for Therastes gaspereti. Here in milder colors you can see the macrochromosomes, and in darker colors you can see the microchromosomes, and in gray you can see the sexual chromosomes. If we see the completeness of this genome, we can see that we got a 92.8% of completeness for Busco. And then if we compare the metrics of our genome, is here, Therastes gaspereti, to other high-quality chromosome-level reference genomes, one of a rattlesnake, one of a cobra, and one of a lizard, we can see that our scaffold M50 is quite high, 214 megabase pairs, in line with the other high-quality reference genomes. In bold is always the best parameter for each of the metrics. The scaffold L50 is the lowest, together with the rattlesnake and the cobra, and the Contic M50 is not the highest, but is quite high, is 45 megabase pairs, almost in line with the genome of the rattlesnake. After that, what we did is to compare the chromosomal rearrangements between the same four species. So in green we have the lizard, in black we have the cobra, in blue we have the rattlesnake, and in orange we have our species, Therastes gaspereti. And in black we have highlighted the chromosomal rearrangements. So we can see that there are quite a lot of chromosomal rearrangements between lizards and snakes, and this is expected due to the innovations that the snakes suffered or experienced during their origin. Then we can see some fusion and fission events between elapids and vipers, but interestingly, we don't see any chromosomal rearrangement within vipers. So this would complete the first objective, that was to produce a chromosome level reference genome. But as I said before, we also were interested in studying the venom. So for that, the first thing we did is to produce the proteome of one individual, and we saw that more than half of the venom is composed only by SBSPs and SBMPs, toxins that are one of the most common toxins within vipers. But what we wanted to do is to identify the genomic location of these toxins within our reference genome. And for that, what we did is to combine several lines of evidence, as for example, proteomics, IsoSeq, RNA-seq, public databases, or homology-based softwares, and we managed to obtain the genomic location of the different toxins. Once we obtained their location, we focused on SBMPs and SBSPs because they were the most abundant toxins within the proteome, as well as phospholipases A2, because they have been widely studied, in order to study them at the genomic level. So what we did is to study them comparing gene copy number variation. Here, this would be the first approach, and also we studied them using a phylogenetic approach. Each row represents one of the different toxins, but today, for simplicity, we're only going to focus on SBMPs. So for SBMPs, we are comparing here. The first species is a cobra, the second is a rattlesnake, and the third species is a true viper, our species Therastes gaspereti. Let's start with the gene copy number variation. And here you have to forget these genes because these are only flanking genes, so they are not toxic. And you can also forget this one here, that is ADAM28, that is the non-toxic paralogous. This means that this gene is not expressed in the venom gland and is not present in the venom, but is highly similar to all the other genes that are venomous. So I only want to highlight 4 interesting things here. The first interesting thing is that if we see all the genes that are within elapids are unique to elapids. And how do we know this? Because when we study them at using a phylogenetic approach, we saw that the elapids were forming an orthologous cluster here that was not shared with any other sample that we incorporated in our phylogeny. As you can see, the colors that are shown here, that are showing the orthologous groups, are the ones that are here in this analysis. So as I said, these genes were unique to cobras, except this one. This one you cannot see the color, but is an MDC1 gene that is orthologous to this one and to this one. The second most interesting thing was that there was a clear expansion of SBMPs comparing elapids to vipers. In vipers, we have quite a lot of more genes of SBMPs. The third interesting result is that there is a second expansion of the SBMPs in rattlesnakes. You can see all these genes that are colored in brown are unique to rattlesnakes. But the fourth and the most important result for us is that this gene here, found in Therastes gaspereti, is found here in the phylogeny and is not showing any orthology with any other gene incorporated in the phylogeny, meaning that this gene is so far unique for Therastes gaspereti. This is possibly due to the lack of sampling that there is within the Viperinea subfamily, because there's not a lot of genomic resources available for this species, and most likely following studies, hopefully or not, will increase the knowledge of this SBMP gene. So to conclude, we have managed to assemble a high quality chromosome level reference genome that is highly contiguous, and in fact is one of the first ones within the subfamily Viperinea. There is a clear absence of chromosomal rearrangements within vipers, and we also want to highlight the importance of using genomics in studying venom evolution, as thanks to genomics we have been able to identify a new gene within our target species. If you're more interested in this article, you can check our preprint that is already available in VR archive. And I would like you to thank you for your attention, as well as the funding bodies.
Okay, thank you everyone to join the session today. So right now we have all finished all the speaker presentations, and we'll the question part and the leave sections and the questions, but what about your thoughts about this part? Richard, can you hear me?
Sorry, yeah.
So now we finished the nine speaker section. Thank you very much. I mean, allow us to thank all the nine speakers for the very good talk, as well as the organizers for having a great event. So now we are going to the another section to which Mark is going to have some music, right? So yes, please, we are going to the next section. Oh, okay. So, sorry. Okay. So this is going to be the Q&A section now. All right. Sorry, I get it confused. So now we are going to the Q&A section. So no, that's fine. We have a lot of questions, interesting questions in the chat box. So Feng and I will be asking. So I just start with what I see on the first one. So for Professor Kuraku. So I didn't quite follow the X chromosome origin story. Did you say that all the elasmobranchs seem to share the same ancestral eggs? Is it reasonably stable in terms of gene content? Do we know the dosage compensation mechanism in sharks?
So, do you hear me?
Yes, we do. Yeah.
Okay. Yeah. Thanks for the question. Overall, the elasmobranchs, I mean, sharks and rays genomes are evolving really slowly, I think. And then, yeah, we haven't compared many species yet, but yeah, sharks and rays seems to share the same, autosomal origin of the X chromosome. And then this is purely based on the, synteny, comparison, focusing on protein coding genes. I think that's a kind of, the only evidence we can really respect in this context. Yeah. Then, yeah, our study and also a study by the Chinese group led by Lo Hao, and yeah, their study also showed the common origin, and both studies showing only little regulation of the expression level between X chromosomes when we compared male and female. So we didn't see a really strong evidence of an dosage compensation, maybe like in birds, I would say. But that study is just done at the transcriptional level, so more studies will be needed, I think. Yeah. Jerome, you are muted.
Okay, sorry. Yeah, it's a very good answer. I think the microchromosomes are also very interesting, but I will go to the next question or in the chat box first. So, this is a question for Richard. So, thanks for your talk. Basic technical question: if you evaluate ONT reads alone, do you see frequent errors at the homopolymers, as known for a long time, I guess, or it performs much better now? Is there a reason why you mainly use Hi-Fi as your basic strategy? So if you can answer that, would be grateful. Yeah.
Sure. So to answer the first part, we've not explicitly looked at the homopolymer error rate in the data. We could do certainly in the past for both PacBio and Nanopore, and I've found in my genome assemblies that most of the errors that certainly make it through after polishing tend to be in the homopolymers for both technologies. I think it has got a lot better. It's still a problem, but I think the problem is getting better, as in you need longer and longer homopolymers for it to be an issue. I know that they're targeted at humans at the moment, but Nanopore is bringing out I think it's G's and C's or G's and A's or something. They have a base modification method that for half the bases they sort of introduce variants of the nucleotide, which means that you get a different signal, so they can actually read homopolymers. But I don't think they've got that rolled out for all 4 bases yet. In terms of why hi-fi, so the Ocean Genomes is aligning with the Vertebrate Genomes Project strategy and trying to basically be as consistent as we can, so that all the genomes that come out are produced in a very consistent fashion. So when all the comparative genomics and things downstream are done, those initial versions of the genome are all sort of put together in the same way. And their main strategy is hi-fi plus hi-C. So that's the one that essentially that we've implemented to start off with.
Okay, maybe I can move to the next questions. And the next question is from the app here, so the Jelco. And how do you identify the mitochondrial and chloroplast genomes in these plants? Okay, everyone here.
I think Jaka didn't get the didn't seem to receive the email, so he's answered in the
yes.
I think he's answered in the Slido.
So I think there's a question for Professor Stu. Why do you think there are fewer SVs affecting introns, or this counting SVs entirely within an intron? So the size could be a factor, or did it include any SVs overlapping introns?
Yes, so that is a good question. There's a couple of reasons. It could be using our method of determining which SV fell into which category. We simply did it, if it touched a gene, then it fell into the gene category. If it didn't touch a gene but touched an intron, it fell into it. So I think there was maybe a little bit of sort of like bias there. But there is also really cool research out there showing that the boundaries between genes and introns have very interesting selection dynamics for and against particular types of structural variants, specifically insertions and deletions. And our data set was very deletion heavy, so we might be seeing then a bias that is specific towards deletions. The reason we have a lot of deletions is that's what short read technology can identify well, and we just went forward with short read technology and the structural variants that we could be very certain about, so we curated them. Yeah, thanks.
Yeah, okay, we can move to the next questions. And is wondering whether your PacBio data was hi-fi or CLR, as I was a bit surprised or how poorly the hi-fi assembly seems to be performing. Yeah.
I'm not sure she's on the call either, is she?
Yeah, but Xiaopeng, Jacko is here, so you can.
Yes. Yeah. Sorry. Yeah. Apparently,
okay. Yeah.
The email was in the junk mail, so now I got the link from Rich. So, yeah. Thanks. So you had some questions now about so there was a question about the chloroplast. So we did assemble those and estimated also the phylogeny there. So the we saw basically the same thing as Zuntini et al. In this Nature paper this spring. So there's an incongruence between the nuclear genome and the chloroplast genome, and then it's because of the ancient chloroplast capture event. So we basically replicate the Zuntini et al. Phylogeny. Then there was a question also about the polyploidy. So it's more prevalent in the boreal and temperate regions, and then in tropics apparently it's not as prevalent. And then we also tested whether there's some genomic plasticity there. So whether these polyploids would be more widely spread than the diploids, and we didn't see any kind of advantage for the polyploids. So the ranges were overlapping, so no significant differences. So apparently this tropical region is just so different. So there's so much competition that the polyploids don't really prevail there.
Nice. So there's an other question for Emma. So mainly asking for your comments. So good work for the RNA part. Don't forget to do native RNA-seq. That's ideal for capturing base modification. For the immune genes, it is genetic recombination signatures that are pretty clear from your genome assembly still, but you never know.
Yeah, thank you. Thank you so much for the suggestion. We're not currently doing native RNA-seq, but we do have access to an ONT Promethean in the lab. So it is definitely something that we could look into. I've also been thinking about the best tissues for sampling for RNA-seq, as gene expression can be tissue dependent. Ideally, we would collect a range of immune organs and perform RNA-seq on more than just one tissue. But this, depends on what we can sample opportunistically. But yes, thank you for the comment. I appreciate the input.
Thank you. So I think most of the questions that we see on the chat box we have asked. So if I may, I have one more question for Professor Kuraku. So for the microchromosomes, I mean, so how often do you see microchromosomes will form? And also, are there different types of microchromosomes? I mean, they will be able to identify the way to which they form. I mean, are there any trend that you still during animal evolution or maybe sharks and rays?
Well, in general, sharks and rays have wide variety of lengths of chromosomes within a karyotype. So it's very difficult to divide between microchromosomes and others, like in birds and reptiles, I think. Yeah, I mean, for example, microchromosomes, or if I use the word microchromosome, the chromosome hosting, or smaller chromosomes in the first species actually is homologous to a very small chromosome in different shark or ray species. So there's a high correspondence between different species, but that's what I can say. And then, if your question about if we can categorize into several types, I have no idea at the moment. And then,
Okay.
So
thank you.
We just sort them with the lengths.
Sure. Okay. Thank you. Right.
So can I ask a quick follow-up on that? Do you see on the contact maps, do you see anything different between the ones that you're sort of classifying as in the microchromosome set? Because certainly when we've done our snake genomes, we see the microchromosomes form just a block altogether, and then there's the other chromosomes. So do you see anything like that with any of your species, or are they all just the same?
Yeah, good point. I think what you say, the block is like an class of chromosomes with the higher chromatin contacts, right? For sharks and rays, we actually do not observe a pattern like that so far.
Okay. So if we don't have further questions, once again, I think we must thank these 9 wonderful and distinguished speakers for giving talks. I must, of course, thank Xiaofeng doing this together, and of course the whole organizer for making this happen for everyone. So this concludes our session for the Asia or Australasia Day for this session. Thank you, everyone.